The National Institute on Deafness and Other Communication Disorders. So if you have a voice, speech, or language project, and you want to submit to NIH, this is one of the major centers that, or institutes that you would send it to. And certainly for voice, this is the place where you send it. Dan.
Chris, do children, infants with this problem, do they speak later if they don't have the elastin gene? Is this not just a voice quality problem, but is there an actual speech problem?
In individuals with Williams syndrome,
they babble later and eventually talk later?
In individuals with Williams syndrome, yes, but not because of the elastin gene abnormality. Because individuals with SVAS have typical developmental profiles as it relates to communication, okay, as it relates to motor abilities. So in that disease, it's just one gene that's missing or irregular, right? But in Williams syndrome, there are multiple genes. So they have the same phenotype, if you will, as SVAS as it relates to elastic fibers and clinical presentation. But there are many other genes which are responsible for the developmental problems we see in Williams syndrome.
That was my next question. Is this related to motor skills at all? In other words, if there's a delay, is there a delay in motor activity?
Certainly that's a factor that could influence whenever you measure individuals with Williams syndrome. You know, the data that you would get as it relates to voice production or muscle tone or really muscle function. Because hypotonicity is a characteristic, is a phenotype of Williams syndrome. Right? So in humans I've studied both, and as it relates to vocal function, I see really the exact same thing. So that, I guess, SVAS are somewhat of a control for Williams syndrome in that case, but I still can't rule out that what I'm hearing and measuring in Williams syndrome isn't due to something else other than just elastic fiber abnormalities, right? It could be muscular abnormalities, it could be, you know, neuron abnormalities, because all of those do happen in Williams syndrome. But just having the SVAS is really a good control for that. And this mouse model is just the SVAS model, so just one gene that's abnormal.
Okay. The mice that are young and old, was that really 5 months to 18 months? Or is that months in the standard deviation and it's 5 years to 18 years?
No, because mice will live 2 years, you're a really old mouse.
Okay, so my question then is, do you already know, and you may have said this and I just missed it, but do we already know that the old mouse does have healthier elastin, or does it digress in them also, like it does a human? So you said that in a human, by the time of puberty, we don't produce any more of the elastin gene, right?
The elastin gene is turned off.
Right.
And the elastin gene encodes the production of elastin, which is the prime, not the only, but the prime constituent of elastic fibers.
So to keep our voices going, if it kind of turns off, then we just kind of go down in voice production from that point. But what about mice? Do you know that they have more of that? Does that elastin gene keep going as they grow older, or do we know that? Is that something you're going to have to check on?
We're not 100% sure, and we're not measuring expression levels of the elastin gene itself, because if it is turned off, we're not going to find anything, right? So we're measuring other genes which are active in elastic fiber formation, but they're also involved in other protein formation too, right? So it's kind of an indirect assessment as to whether or not there's more activity going on in the genes. But, you know, this has never been studied before, so we're not quite sure what is going to happen. Although, if you look at other genes, like collagen genes, right, in young and old mice, young and old rats, there are differences in activity levels of expression in those genes.
Okay. So it may be that you find similar results, but you're not even sure of that at this point.
Correct. We just have a hypothesis.
And if that's the case, then you're going to be looking at how we can produce or manufacture.
It's the Holy Grail, right? I mean, if people very, 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 like Nobel Prize winning people are in the world today working on this problem of how do we turn that elastin gene back on? Because now you're going to cure aging in a lot of tissue for a lot of humans, right? Well, theoretically. How the elastin gene functions in the vocal folds, we really don't know. And so this is generating some evidence which helps us understand that. Hopefully down the road, once we do understand the elastin gene, how it functions in health and disease and aging, then we can, once we have that knowledge, then we can start addressing issues related to treatment.
And in the comments of the people from the grant group, did they like the direction you're going? It's just that they ran out of money to fund
I can't believe I ended up getting the score I got, because the 3 reviewers who initially reviewed it before the study group gave me 1s. 1 is the highest score, 9 is the highest. 9 is the lowest score you can get. It wasn't that good. 1 is the best score you can get. I got like 1s and 2s for most. And then when it went to study group, obviously there were some people who weren't as high on it. So I ended up getting a score of 5.9, which is kind of mid-range. So obviously there were 4 studies that were rated higher than mine that got funded. But the main objections were to the approach as it applied to the acoustics. They wanted me to do a different type of analysis, which would translate better to measurements that are taken in humans. So they wanted a textual analysis of the acoustic spectrum. They said that the analyses we were going to do really clinically would have no great translation. So I'll be able to do these.
That was pretty much.
That was really the major concern. I got really good markings. I know some of the people who are in that study group. I'll see them next month. I want to know what happened inside that room. But there's just so little money. There's not. It's so tight. And there are some great people, much greater and smarter than me, applying for the same money. So it's just really difficult. And also, they're part of the club, I'm sure.
Yeah.
Trying to work on the club.
Tell us what an R21 is.
It's a developmental and exploratory grant, so it will fund up to, not including indirect costs, upwards of $275,000. I think the total of this grant was about $370-something thousand with indirects. And the purpose of that is to fund research which explores novel methodologies which should lead to larger projects that are funded by an R01. Okay? So R01 could be up in the millions of dollars. So the R21 allows researchers to, on a smaller scale, develop lines of research and methodologies which will substantiate another larger application for future research.
And tell us what mice cost.
They're not that expensive.
They're not?
No, you can, from Jackson Laboratories, you get a mouse for about $10. What's expensive is taking care of the mice. But TCU is a special place in many ways. Science and engineering has a vivarium paid for by the college that they allow individuals to house mice. Now, I'm lucky in that respect, but I'm also doubly lucky in that all I need is the larynges of these mice. And so my colleagues at Washington University in St. Louis who study these, who use these mice to study the cardiovascular defects that happen in SVAS, they don't use the larynges. So I'm able to get the larynges, let them do all the husbandry and the hard work. All I have to do is cut the larynx out and bring them back to TCU.
Ice them down.
Ice it down.
So would you have had to bought the mice and let them age 18 months? Or would they, you already had the ones you
I don't have a colony here yet. The idea is to get funded, but they have these mice up there. And so as the study progressed, I would get a breeding colony, establish that colony here, but at the same time I'm collecting data on mice that are from the same colony, the original colony, that are at Washington University in St. Louis. So I have little mice running around in the lab. Not in Miller.
Little colony. All right, well, thank you.
Okay, thanks.